We have three big intertwined systems on the table today. Smooth muscle, the massive cardiovascular network, and the absolutely crucial lymphatic drainage system.
And if we really want to get a grip on how the body manages all of this, volume, pressure, velocity, everything from the high-speed jet of blood leaving the heart to the slow trickle in your capillaries, we have to start with the master regulator.
Right.
And that's smooth muscle.
Right.
It really is the unsung hero of all involuntary movement, and it lines pretty much every tubular structure in your body.
I think we really do overlook smooth muscle. I mean, it's not the glamorous bicep or the tireless heart muscle, but its versatility is it's astonishing.
Yeah.
So our focus starts here because this tissue is found, like you said, everywhere. It's in the walls of every blood vessel, well, outside of the smallest capillaries. It's in the digestive tract, the airways, even the tiny muscles that make your hair stand on end.
It's the tissue of adaptability.
Uh-huh.
And the fundamental difference that sets it apart right away is structural.
Okay.
Unlike skeletal muscle, which, is characterized by those visible stripes or striations.
Yeah, the ones you see in textbooks.
Exactly. Smooth muscle lacks that regular organization of contractile proteins into those repeating units we call sarcomeres. And this absence of visible stripes is, well, it's why we call it smooth.
So, okay, if it doesn't have those neatly organized A bands and I bands and Z discs that create that sort of linear pull we associate with, lifting a weight, how does it still contract with so much force and efficiency?
That's the big question.
That seems to be the foundational question we really need to answer here.
And it achieves that force generation through a completely different, almost a nonlinear architecture, which we'll definitely unpack in a minute. Functionally, it is, as we said, generally involuntary.
Right. Thankfully.
Thankfully, you don't have to consciously regulate the tone of your arterioles to maintain your blood pressure. Control is handled by the autonomic nervous system or by local hormones, even paracrine signaling from nearby cells, which gives it this incredibly flexible, localized control.
What's really fascinating, and something the sources definitely highlight, is that smooth muscle isn't just one single thing. It seems to adapt its connectivity based on what its job is.
Precisely. We see two main functional types. The first is single-unit smooth muscle. This is where the cells are extensively connected by these things called gap junctions.
Okay, gap juncture.
And these allow them to contract rhythmically and simultaneously as one single electrical unit. A functional syncytium is the technical term.
A syncytium, so like a single coordinated sheet of muscle.
Exactly. And this is what you see in the gut or the uterus or in small blood vessels where you need these coordinated propagating waves of contraction.
Okay, so that's single unit. What's the second type?
The second is multi-unit smooth muscle. Now here, the cells are often innervated individually.
So much more precise.
Much more precise. It allows for very localized control. The muscle cells can contract independently of their neighbors. And you see this in structures that need really fine adjustments, like the muscle that controls the pupil of your eye, or in certain large elastic vessels that require more localized tweaks to their tension.
Let's get microscopic for a second. Can you describe the basic cell structure? I mean, they must be quite different from those long, cylindrical, skeletal muscle fibers we usually picture.
Oh, they're drastically smaller and very distinct. Smooth muscle fibers are spindle-shaped, so they're thick in the middle and they taper at both ends, and they're comparatively tiny. We're talking 20 up to maybe 500 micrometers long, but only 3 to 8 micrometers in diameter.
That's incredibly small.
It is. And they're almost always arranged in layers or bundles. Typically, you'll see an inner layer that's oriented circularly and an outer layer that's longitudinal, especially in the walls of the gut or large vessels.
And the nucleus. I remember from histology that the nucleus in smooth muscle is often the giveaway under a microscope, isn't it?
It is the defining feature, absolutely. Each cell have a single, centrally located nucleus. But here's the visual trick.
Okay.
When the cell is relaxed, the nucleus just looks like a simple oval. But when the cell contracts and shortens, that internal shortening and twisting of the cell body forces the nucleus to wrinkle up. It takes on this highly characteristic, almost diagnostic corkscrew shape.
A corkscrew.
Or like a concertina, being squeezed.
Yeah.
It visually confirms the cell is actively shortening.
That twisting action, it sounds like that's the key to understanding how force is transmitted. If these tiny individual cells need to work together as a synchronized layer like they have to in a blood vessel wall, how do they actually link up, both mechanically and electrically?
Right, so they employ a pretty sophisticated connection system to get this coordination. Mechanically, they're linked by what are called intermediate junctions. And these resemble desmosomes, which you might be familiar with. They're strong, localized sites where the cytoskeletal networks of adjacent cells are basically fused to the membrane. This allows the physical force of contraction to be transmitted really efficiently from one cell to the next, right across the whole tissue layer.
Okay, so that's the mechanical, the physical pulling.
hmm
But for the required rhythmic movement, like, say, peristalsis in the gut or that simultaneous constriction in an artery, you need the electrical coordination too.
That's where the gap junctions are absolutely vital, particularly in that single-unit smooth muscle we mentioned.
Right.
These are specialized areas of the cell membrane that create these low-resistance connections. They're essentially tiny tunnels between the cytoplasms of neighboring cells.
So things can pass right through.
Ions can pass directly through these tunnels.
Yeah.
This allows depolarization waves to spread almost instantly across the entire sheet of muscle. This electrical coupling is what ensures the whole muscle mass contracts almost simultaneously, fulfilling that requirement of a functional syncytium.
So we have these small spindle-shaped cells, and they're linked up both electrically and mechanically. Now let's tackle that central architectural challenge you mentioned. How do you get a powerful contraction without the linear efficiency of Z disks? How is that force actually managed?
The answer lies in this really unique internal scaffold and anchoring system, which is far more diffuse than the skeletal muscle model. The core anchoring structures are called dense plaques and dense bodies.
Okay, let's define those two. They sound important.
They are. So dense plaques are found right underneath the cell membrane. They act as attachment points that are integrated right into the plasma membrane. You can think of them as functionally analogous to Z-discs because they are the attachment sites for the contractile filaments.
Okay, so they're on the edge of the cell. What about the dense bodies?
The dense bodies are very similar in composition, but they're found scattered throughout the cytoplasm, deep inside the cell.
Ah, so it sounds like an internal three-dimensional lattice rather than a stacked linear arrangement.
Precisely. That is the perfect way to describe it. These dense bodies and plaques serve as anchoring points, not only for the bundles of actin and myosin, but also for this robust intermediate filament network.
Okay.
This network, which is composed primarily of proteins called desmin and vimentin, forms the internal skeleton of the cell. So when the cell gets the signal to contract, the force that's generated by the sliding filaments is transmitted through this entire desmin and vimentin lattice, and it pulls all of the numerous scattered dense bodies and plaques inward.
And that pulling action is what causes the entire cell to sort of bunch up and shorten, almost like wringing out a wet cloth, which I guess explains that corkscrew nucleus you were talking about.
It does.
It's an incredibly efficient way to achieve shortening across multiple axes at once.
It is a remarkable kinematic solution. And the filaments themselves, they're optimized for this sustained ringing action. Smooth muscle actin filaments are actually much longer than those in skeletal muscle. We're talking up to 2 micrometers compared to just 1 micrometer.
Wow, double the length.
And here's where the mechanical ratio really changes. The ratio of actin to myosin is much higher. It's approximately 15 to 1 compared to about 2 to 1 in skeletal muscle.
15 to 1, that's a huge shift in composition. I mean, that's a lot more surface area for contact. What is the functional consequence of having that high ratio and these longer filaments?
It contributes significantly to what we call the power factor. Smooth muscle can generate a substantial amount of force per unit cross-sectional area. Often, and this is surprising to many people, more force than skeletal muscle.
More? Really?
Yes, because the smooth muscle myosin filaments are also longer, and they can interact with a greater proportion of those long actin filaments for a much more extended period of time. The entire system is designed for prolonged, highly economical tension.
So the overall mechanism is it's slow to start, but it's incredibly powerful, sustained, and highly energy efficient. This is the biological requirement for constantly maintaining vessel tone, isn't it?
It is the perfect adaptation. This efficiency allows smooth muscle to sustain what we call tonic contractions, like the constant pressure needed in an artery wall, for hours or even days with minimal ATP consumption.
How does it do that?
It utilizes a special state known as the latch state, where the myosin heads, once they're attached to actin, detach very, very slowly. This allows the muscle to maintain tension using very little energy. It's a biological marvel of energy conservation.
A latch state. I like that. Okay, now that we understand the internal motor, let's explore the regulatory system. This tissue operates completely outside of our conscious control, relying on the autonomic nervous system. So how is that connection made, given that it doesn't seem to need that precise, tight synapse you see in skeletal muscle?
Right, the connection system is structurally very distinct. The innervation is sparser, and the vessels that supply the muscle, the capillaries, they often don't run in direct relation to individual cells, unlike in striated muscle. The mechanism is really one of diffusion and wide area control.
Okay, tell us more about that diffuse control system. That sounds interesting.
We refer to these as autonomic neuroeffector junctions. So instead of a single defined neuromuscular junction like in your bicep, the nerve endings, which are part of the sympathetic or parasympathetic divisions, they contain these swellings along your length called varicosities.
Varicosities, like little beads on a string.
That's a great analogy. And these varicosities are packed with neurotransmitter vesicles.
So when a signal arrives, it doesn't just go to a single point on a single cell, it's distributed.
Exactly. When an action potential reaches the nerve ending, the neurotransmitter is released from many of these varicosities all at once. And critically, these nerve endings are often separated from the smooth muscle cells by a pretty wide gap, anywhere from 10 to 100 nanometers.
Wow.
So the transmitters just diffuse across this distance, and they influence many adjacent muscle cells simultaneously. This enables these widespread, coordinated changes across an entire sheet of muscle, which is precisely what you need to regulate the radius of a blood vessel.
And the sources also mention dual control, which suggests there's a complex push-pull system regulating this diffusion.
Yes. Most visceral, smooth muscle is innervated by both sympathetic, which is often excitatory, causing constriction, and parasympathetic, which is often inhibitory, causing relaxation divisions.
Ah, so you have a gas pedal and a brake.
You do. And this dual input allows for highly nuanced control over functions like vessel diameter or airway resistance or gut motility. The specific response, excitation or inhibition, it depends entirely on the type of receptor that's present on the smooth muscle cell membrane.
That brings us to the ultimate trigger, the calcium dance, which is what actually regulates the contraction. Since it doesn't use troponin like skeletal muscle, the mechanism must rely on a different set of cellular switches.
It does. While the contraction can be initiated by all sorts of stimuli, electrical changes, mechanical stretching, chemical signals like hormones, all roads lead to an increase in intracellular calcium. And the key point here is that smooth muscle relies significantly on extracellular calcium influx.
So unlike skeletal muscle, which mainly just relies on its own internal stores in the SR, smooth muscle is actively pulling a lot of its triggering calcium right out of the blood plasma through the cell membrane.
Correct. This influx occurs through various channels that are opened by electrical changes, which are voltage-gated channels, or by specific signals like neurotransmitters or hormones, which are receptor-operated channels. And although there is some internal release from the sarcoplasmic reticulum via ryanodine receptors, the dependency on that external calcium is far, far greater than in striated muscle.
Okay, so once that calcium arrives inside the cell, how does it physically activate the myosin head? I mean, we need a really clear comparison here to the troponin system that we're more familiar with.
This is where the regulatory pathway just diverges completely.
Yeah.
Smooth muscle does not use troponin C. Instead, the incoming calcium binds to a ubiquitous regulatory protein called calmodulin.
Calmodulin.
Yes. And this binding forms the calcium calmodulin complex.
And that complex, that's the key switch.
It is the ultimate activator. The calcium calmodulin complex then goes on to activate a specific enzyme, myosin light chain kinase, or MLCK for short.
MLCK.
And MLCK's entire job is to use ATP to phosphorylate, which just means to add a phosphate group to the regulatory light chain of the myosin head.
So phosphorylation is the green light for contraction.
Absolutely. The phosphorylation unlocks the myosin head. It allows it to bind to actin and initiate that cross-bridge cycling and force generation. If the myosin is not phosphorylated, it remains detached and the muscle is relaxed. This is why MLCK is the critical regulatory step for initiation.
Okay, that makes sense. So what stops the process? What's the relaxation mechanism that lets the cell go back to its resting state?
Relaxation requires deactivation. Specifically, it requires dephosphorylation. This is handled by a different dedicated enzyme called myosin light chain phosphatase, or MLCP.
The phosphatase, it takes the phosphate off.
Exactly. MLCP removes that phosphate group from the myosin light chain, and that causes the myosin to detach from the actin, which leads to relaxation.
So if MLCK initiates and MLCP ends the process, which one dictates the speed of the whole cycle?
It's generally considered that MLCP's activity is the rate-limiting step for relaxation in smooth muscle. Because that dephosphorylation step is slow, it helps explain the much slower kinetics of smooth muscle contraction and relaxation compared to the almost instantaneous action of striated muscle.
And that slow kinetics is, again, perfectly tailored for maintaining that sustained vessel tone with incredible energy conservation, that latch state we mentioned.
That's right. It all ties together.
That constant low energy presence is just so vital. Finally, before we leave the cell itself, what about its capacity for repair and change? The source material notes its really remarkable plasticity.
Right. Most smooth muscle originates from mesenchymal sources during development. What's exceptional about it is that smooth muscle cells retain their capacity for growth and adaptation throughout life. They can undergo hyperplasia.
Which is multiplication of cells.
Yes. And hypertrophy, which is the enlargement of the existing cells.
Like the massive and temporary growth required in the uterus during pregnancy. That's a perfect example.
A classic example of adaptive hyperplasia.
Yeah.
But this plasticity has a pathological dark side, particularly in the cardiovascular system.
How so?
Smooth muscle cells are capable of switching their phenotype. They can actually change from being contractile cells to being synthetic cells, which produce large amounts of extracellular matrix proteins, like collagen.
And how does that relate to disease?
This phenotype switch is heavily implicated in vascular remodeling and pathologies like atherosclerosis. The smooth muscle cells actually migrate into the tunica intima, they proliferate there, and they contribute significantly to the fibrous cap of atherosclerotic plaques.
So they're part of the problem.
A huge part of the problem. And a lot of current therapeutic efforts are, in fact, focused on finding ways to reverse these maladaptive remodeling pathways in vessel walls, trying to turn them back into their healthy contractile state.
Wow. Okay, so that foundational knowledge of smooth muscle really allows us to appreciate the mechanics of the plumbing system itself. In part 3, let's zoom out to the sheer scale and the dual loops of the cardiovascular network.
The scale is indeed staggering. We're talking about a closed two-loop circuit that's powered by the heart, which is acting as two parallel pumps, managing two wildly different pressure systems all at the same time.
And the two loops are the pulmonary and the systemic circuits.
Right. The pulmonary loop is the low-pressure side. Its sole purpose is to circulate blood through the lungs to exchange carbon dioxide for oxygen. The right side of the heart is the pump for this loop.
Low pressure makes sense. It's a short trip.
Exactly. Then you have the systemic loop. This is the high-pressure, high-resistance circuit powered by the mighty left ventricle, and it's designed to distribute that newly oxygenated blood to every organ, tissue, and periphery in the entire body.
And this inherent difference in pressure, low pressure for the lungs, high pressure for the body, is exactly why the left ventricular wall is so much thicker and more muscular than the right.
Precisely.
But let's talk about that critical pressure gradient as blood leaves the high-pressure aorta.
The pressure dynamics are absolutely crucial as they dictate the structure of every single vessel downstream. Pressure starts extremely high, your systolic pressure in the aorta. It drops steadily as blood moves through the massive conducting arteries, but the steepest, most dramatic drop occurs as blood navigates the tiny arterioles.
And by the time it gets to the capillaries, the pressure is very low.
Very low.
So why is that massive pressure drop in the arterioles so essential?
It serves two main purposes. First, it converts that pulsatile flow, the surges from each heartbeat, into a smoother, continuous flow that the very delicate capillaries can actually handle.
Ah, okay.
And second, the lower pressure and velocity in the capillaries are absolutely essential for facilitating exchange. If the blood moved through too quickly, there just wouldn't be enough time for gases and nutrients and waste to diffuse across the capillary wall.
Speaking of flow, the geometry of the system is, well, it's bizarre when you think about it. The aorta is relatively wide, maybe 30 millimeters in diameter, but when you aggregate the cross-sectional area of the final destination, the entire capillary bed, the change is massive.
It's a remarkable feature of biological engineering.
Yeah.
While the aorta is a single narrow pipe, the total cross-sectional area of all the capillaries combined is estimated to be around 2,500 times greater than that of the aorta.
2,500 times?
Yes. And this vast increase in area causes the blood flow velocity to decrease dramatically. It slows down to a snail's pace, which maximizes the time available for molecular exchange at the tissue level.
So velocity is inversely proportional to the total cross-sectional area. That ensures that gas diffusion has enough time to actually happen. That's really elegant.
It is.
Okay, now let's look at the standard vessel structure. The source material talks about the three tunics, which seem to define the composition of every major vessel wall.
That's right. Regardless of whether we're looking at a large artery or a medium-sized vein, most vessels share this common concentric layered plan. Starting from the inside, we have the tunica intima,
This is the innermost layer, the one that's in direct contact with the blood.
Yes, and its most critical element is the endothelium, a single, flattened layer of specialized squamous cells. Now, this layer is far more than just a simple lining.
It's active.
It's highly metabolically active. It forms a non-thrombogenic surface, so it prevents inappropriate clotting. And it releases crucial signaling molecules like nitric oxide, which causes vasodilation, and endothelin, which causes vasoconstriction. So it's directly regulating the smooth muscle in the layer below it.
So it's the ultimate boundary layer, regulating the flow chemistry. That's fascinating. Okay, so below that, we hit the heavy engineering of the wall, the tunica media.
The tunica media is the middle layer, and it's the functional determinant of the vessel. It's primarily composed of those circumferentially arranged, smooth muscle cells we've been talking about, and varying amounts of elastic fibers. This is the layer that dictates the vessel's compliance, its ability to recoil, and its resistance. Its muscle content is what allows the vessel to actively regulate its own diameter.
And the final, protective jacket on the outside.
That's the tunica adventitia, the outermost layer. It's primarily connective tissue, mostly collagen, which provides structural containment and anchors the vessel to all the surrounding tissue.
Okay.
And in larger vessels, where the wall itself is too thick for oxygen and nutrients to diffuse from the blood flowing inside, the adventitia also houses the vasa vasorum.
The vessels of the vessel.
Literally the vessels of the vessel. They're just small arteries and veins that supply oxygen and remove waste from the muscle layer itself.
Okay, so let's use those tunics now to differentiate the high-pressure arteries. In Part 4, the source material highlights two very distinct functional classes, and it's based on the composition of their tunica media. We should start with the giants, the elastic arteries.
The elastic arteries are the largest vessels. We're talking about the aorta and its immediate main branches. Their primary job is not to regulate flow to specific organs, but to manage the immense pressure surge that's coming directly from the heart.
So they are the ultimate pressure buffers, the shock absorbers.
Absolutely. Their tunica media is characterized by the dominance of elastic fibers over smooth muscle. They're arranged in 50 to 70 concentric sheets or elastic lamellae. These vessels are stiff yet compliant, which is a perfect structural adaptation for absorbing that shock.
And this leads to that remarkable physical principle known as the Windkessel effect. Can you walk us through the genius of that mechanism?
Sure. Think of it like a surge protector, but for hydraulics. During systole, which is the phase when the left ventricle ejects blood under high pressure, the elastic arteries expand almost instantly. They accommodate a significant portion of that stroke volume.
They stretch.
They stretch. They act like compressed springs, temporarily storing the mechanical energy of that ejected blood.
And then what happens when the heart enters diastole and the ventricle relaxes?
During diastole, the pressure drops sharply at the heart, but the expanded elastic walls of the aorta recoil passively. All that stored energy is released back into the blood column, pushing the blood forward continuously. This recoil is crucial because it maintains significant blood pressure and flow downstream, even when the heart is relaxed. It smooths out the pulse wave and ensures a continuous, non-stop perfusion of the downstream capillary beds.
So the loss of this elasticity over time, which the sources note happens as elastic fibers fragment and accumulate lipids with age, that's a core reason for age-related systolic hypertension. The surge protector just stops working.
Exactly. Stiffer arteries mean they can't absorb that systolic pressure surge as effectively. So the pressure rises much higher during systole. And because they recoil less, the pressure drops lower during diastole. This increases the amplitude of the pulse pressure, and this mechanical failure places severe strain on the smaller, more delicate downstream arterioles and capillaries.
Wow. Okay, so now contrast that shock absorber function with the muscular arteries. These are the vessels further down the line, the ones actually distributing blood to the specific organs.
Right. The muscular arteries are the gatekeepers of distribution. Their tunica media is overwhelmingly dominated by those circumferentially arranged, smooth muscle cells.
Lots of muscle.
Lots of muscle. They still have elastic tissue, of course, and it's marked by a very distinct internal elastic lamina and an external elastic lamina, which sort of bookend the muscle layer.
So if the elastic arteries manage the bulk pressure, the muscular arteries are managing the localized flow via constriction and dilation.
That is their central role. Their thick, smooth muscle layer allows them to precisely regulate their lumen diameter. And this regulation can be controlled by central sympathetic nervous system input or, crucially, by local metabolic demands.
So for example, if your arm muscle needs more oxygen during exercise.
Exactly. The muscular arteries feeding it will receive signals, often local metabolic ones, to dilate, which can actually override the central tone from the brain.
And that precise control becomes most vital in the smallest of the arteries, the arterioles.
The arterioles are disproportionately important because they are the primary site of systemic vascular resistance.
This is where that huge pressure drop happens.
This is it. They are the smallest arteries, and their walls are very thin, often containing just 1 or 2 continuous layers of smooth muscle cells in the media. But they regulate the entire systemic blood pressure. A tiny change in the radius of the arterioles results in a massive change in total resistance and consequently pressure.
So if we need to regulate blood pressure rapidly, like when you stand up too fast, we target the smooth muscle in the arterioles. What exactly controls that tone?
Arteriolar tone is maintained by a constant sympathetic innervation, a baseline state of partial constriction, which we call vascular tone. Noradrenaline released from sympathetic nerves acts on receptors in the smooth muscle, and this typically causes vasoconstriction.
But it's not just the nerves.
No. This sympathetic command is constantly being modulated by circulating hormones, like angiotensin II, and local metabolic factors. For instance, high CO2 or low oxygen in a tissue will locally trigger a massive vasodilation, proving that local need can often trump central control.
Fascinating. Okay, moving into part 5. We are now entering the realm where the rubber meets the road, the capillaries. This is the critical crossroads where the entire purpose of the circulatory system exchange actually happens.
Right, and capillaries are structurally the simplest vessels, yet they are the functional core of the whole system. They consist only of a single layer of squamous endothelial cells and a supporting basal lamina. That's it.
Thin.
Incredibly thin. And they're small dimensions. We're talking 4 to 8 micrometers. I mean, red blood cells must literally pass through in single file, which maximizes their contact time with the vessel wall and minimizes the distance for diffusion.
And we also see those associated regulatory cells, the pericytes, sitting on the outside of the capillary wall. What's their function?
Pericytes are crucial, multifunctional cells. They wrap around the capillary and the postcapillary venules, and they possess contractile elements, which suggests they may have a local role in regulating blood flow or permeability.
So they can squeeze the capillary?
They can. And furthermore, they're vital in maintenance and repair. They participate in angiogenesis, which is new vessel formation, and they have high plasticity, meaning they can differentiate into smooth muscle cells or fibroblasts following an injury. They're sort of the local maintenance crew.
The sources are really clear on this point. Not all capillaries are created equal. Their specialized types seem to reflect the tissue's specific need for permeability, and we have 4 main functional variations.
We do. We could begin with the most restrictive type, continuous capillaries. They're the most common. You find them in muscle, skin, lung, and nervous tissue. The endothelial cells here are very tightly linked by tight junctions, which creates a continuous, uninterrupted barrier.
So not much gets through.
Very little. Exchange is restricted primarily to the diffusion of small lipophilic molecules or active energy-intensive processes like transcytosis, which is transport via tiny vesicles moving across the cell.
Okay. Next up, the type designed for more accelerated movement, fenestrated capillaries.
Fenestrated capillaries are engineered for rapid fluid and small molecule exchange. Their defining feature is the presence of numerous circular openings, or fenestrations, in the endothelial cells. And in most locations, these pores are actually covered by a thin, non-membranous diaphragm.
Fenestrations, like little windows, where do we find them and what's the functional advantage?
They are abundant in organs involved in high-volume filtration and secretion. So think of the kidney glomeruli, endocrine glands for hormone secretion, and the intestinal villi for absorption. The diaphragm on those pores restricts the passage of large molecules like plasma proteins, but it allows water and small solids to pass through very rapidly, hence their role in quick filtration or absorption.
Okay, that makes sense. The third type seems to represent the maximum level of permeability allowed anywhere in the body.
Those would be the sinusoids, or discontinuous capillaries. They are expanded, irregular vessels. Sometimes they can reach 30 to 40 micrometers in diameter. And critically, they have large gaps and discontinuities between the endothelial cells, and their basal lamina is often incomplete or even absent.
So large molecules and even entire cells can pass through here.
Yes. They are found where maximum exchange or interaction with blood components is needed. The liver, for example, where large plasma proteins are synthesized and must enter the circulation. The spleen, where aged blood cells are removed. And the bone marrow, where newly formed blood cells have to enter the circulation from the marrow space.
And finally, there's the specialized endothelium, which is designed to actively prevent exchange.
Right. This is best seen in the central nervous system, and it forms the basis of the blood-brain barrier, or the BBB. Here, the tight junctions in the continuous capillaries are extraordinarily developed. They limit paracellular transport almost completely.
So things can't sneak between the cells.
Not at all. Exchange is highly restricted to specific, tightly regulated transport mechanisms. And this protected environment, which also involves associated parasites and the end feet of astrocytes, ensures the stability that's required for proper neural function.
Now that we understand the structures that allow for movement, let's discuss the critical physics that governs the movement itself. Fluid exchange as governed by the Starling forces. This is the constant, unseen work that maintains the fluid balance across every single tissue in your body.
This is a perfect example of two opposing sets of forces ensuring biological homeostasis. We're constantly balancing fluid between the blood plasma and the interstitial fluid that's surrounding the cells. The first set of forces is the hydrostatic pressure.
That's the push force.
Correct. Hydrostatic pressure is the force exerted by the blood pressure itself against the capillary wall. This pressure tends to push water and small solutes out of the vessel and into the interstitial space, and it's highest at the arterial end of the capillary.
Okay, so that's the push. What about the opposing force that pulls fluid back in, the osmotic section?
That is the colloid osmotic pressure, or sometimes called oncotic pressure. This force is generated almost entirely by the presence of large plasma proteins, chiefly albumin, which are generally too large to leave the capillary, except in those sinusoids we mentioned. These retained proteins create a concentration gradient that effectively draws water back into the vessel.
So how does this battle play out along the length of a single capillary?
Okay, so at the arterial end, the hydrostatic pressure is dominant. It's high enough to force fluid out. That fluid, which is loaded with nutrients and oxygen, bathes the surrounding cells. As the fluid is filtered out, the remaining blood volume in the capillary decreases slightly, and so the hydrostatic pressure drops significantly along the length of the capillary.
And meanwhile, the colloid osmotic pressure is staying relatively constant because the big proteins haven't left the blood.
Exactly. So by the venous end of the capillary, the hydrostatic pressure has dropped below the colloid osmotic pressure. At this point, the osmotic pull of those retained proteins becomes the dominant force, and it pulls the interstitial fluid, which is now rich in waste products like CO2, back into the capillary.
But the sources seem to indicate that filtration almost always exceeds reabsorption slightly, meaning not all the fluid that leaves at the arterial end actually comes back at the venous end?
You are absolutely right. There is a slight net filtration of fluid into the interstitial space. We're talking about 1 to 2 liters per day across the whole body that is not reabsorbed back into the capillaries.
Okay, so where does it go?
This excess fluid, along with any proteins that might have escaped, must be collected. And that is the job of our third-grade system, the lymphatics.
And when this system fails, that's when we get edema, which is a very visible consequence of this Starling forces imbalance.
Edema is the localized swelling that results from this failure. It can happen if the hydrostatic pressure is pathologically elevated, for example, in heart failure or a deep vein thrombosis, which causes a backup of blood pressure pushing too much fluid out.
Oh.
Or it can happen if the colloid osmotic pressure is too low, for instance, in severe liver failure, where albumin production stops. Then there isn't enough osmotic pull to bring the fluid back in.
Mm-hmm.
In either case, the excess fluid gets trapped in the tissues, and that leads to swelling.
Right. Okay, so in part 6, let's look at these low-pressure return systems. The veins and the lymphatics, they both have to fight gravity and return fluid to the heart without the benefit of that high-pressure pump. Starting with the veins, they are clearly built for volume, not for pressure resilience.
They are the ultimate capacitance vessels. Structurally, veins have much thinner walls and larger, highly distensible lumens compared to arteries of a similar diameter. The tunica media is relatively weak and thin, and this construction allows them to hold a vast amount of blood. They're estimated to contain up to 70% of the body's total blood volume at any given time.
70%? Wow. And while the artery's adventitia is mainly for anchoring, the veins adventitia is often its thickest layer.
That's right, especially in large veins. The tunica adventitia can be quite thick and often contains longitudinal bundles of smooth muscle cells, as well as collagen and elastic fibers. This outer robustness provides the necessary structural support despite the thinness of that medial layer.
Since the pressure is so low, particularly in the limbs, the body needs an active mechanism to ensure blood moves unidirectionally back toward the heart against gravity. This brings us to the venous valves.
Venous valves are absolutely essential, especially in the extremities, but they are absent in the central veins of the chest and abdomen. They're formed by two semilunar folds of the tunica intima, and they act as perfect one-way gates.
So how do they work?
When blood flows toward the heart, the valves open passively. If blood starts to flow backward due to gravity or pressure fluctuations, the cusps fill with blood and they snap shut, which prevents backflow and ensures that unidirectional return.
So the valves provide the directional control, but the movement itself requires some external power. That power comes from two pumps.
The first is the skeletal muscle pump. When you walk or contract the muscles in your legs, the contracting muscles compress the deep veins that are contained within them. This external pressure pushes the blood forward. The valves immediately proximal to the compression open, while the valves distal to it close, which guarantees movement toward the heart.
So that's why walking is so important on a long flight.
Absolutely. It keeps the blood from pooling. And the second pump is related to respiration.
Okay.
It's the respiratory pump or the thoracoabdominal pump. As we inhale, the diaphragm descends. This increases abdominal pressure and at the same time decreases intrathoracic pressure. This pressure difference creates a suction effect, which draws blood from the high-pressure abdominal veins toward the low-pressure thoracic veins and eventually into the right atrium. Both pumps work in tandem, turning muscle movement and breathing into circulatory mechanics.
The microcirculation also uses some structural adaptations for quick flow control, namely these things called arteriovenous shunts and anastomosis.
Right. Arteriovenous shunts are direct, short connections between a small artery and a vein, and they allow blood to entirely bypass the capillary bed.
Bypass it completely.
Completely. They are functionally critical, particularly in the skin of the extremities, so your fingertips, ears, nose.
And what role do they play there?
Thermoregulation. When the body is cold, the sympathetic nervous system stimulates the smooth muscle surrounding these shunts to relax and open up the shortcut. Blood bypasses the superficial capillaries, which minimizes heat loss to the environment.
And when you're hot?
When the body overheats, the shunts constrict, which forces blood through the surface capillaries for heat dissipation, which is why your skin gets flushed.
And anastomoses are the circulatory redundancy backups. They're designed to prevent catastrophic failure if a major vessel gets blocked.
Exactly. Anastomoses are connections between larger arteries. Think of the collateral circulation around major joints or in your hands and feet. They provide alternative routes like off-ramps and bypass roads on a highway, ensuring that if a primary vessel, say your radial artery, becomes blocked, blood flow is still maintained to the downstream tissue via connections from the ulnar artery, which prevents ischemia.
A built-in backup plan. Now let's look at the final piece of this return trip puzzle, the lymphatic system. This network handles that fluid and protein overflow that the Starling forces couldn't recapture.
The lymphatic system is the essential drainage network. Its functions are threefold. Draining that 1 to 2 liters of net-filtered interstitial fluid we talked about, transporting absorbed dietary fats, which are called chyle, from the intestines via specialized vessels called lacteals, and performing immunological monitoring through the lymph nodes.
Structurally, how does it differ from the blood capillaries, and how does it manage to suck in that excess fluid and protein?
It begins as blind-ended lymphatic capillaries in most tissues. They're sacs, not loops. And their cellular structure is unique and really key to their function. The endothelial cells are highly attenuated, they overlap like shingles on a roof, and they lack the tight junctions that you find in blood capillaries.
That shingle structure sounds like a perfect one-way entry valve.
It is. When the interstitial fluid pressure rises, it forces the overlapping endothelial cells inward, and they act like one-way swing doors, allowing fluid, proteins, and even immune cells to enter. And crucially, these capillaries are anchored to the surrounding connective tissue by microfibrils, which ensure that when the tissue swells with fluid, the microfibrils actually pull the endothelial cells apart, holding the channels open and facilitating drainage.
That is so clever. Okay, once that fluid, which is now called lymph, is inside, how does this system, which is also very low pressure, move it all the way back up to the chest?
The lymphatic vessels strongly resemble veins. They have thin walls, large lumens, and numerous valves. And the lymph is propelled by a combination of external compression that skeletal muscle pump acts on lymphatic vessels too, and internal action. The walls of the larger lymphatic vessels contain layers of smooth muscle that undergo their own intrinsic rhythmic contractions.
They pump themselves.
They create a peristaltic wave that pushes lymph forward from one valve segment to the next.
The ultimate fate of this lymph is to return to the blood near the heart to complete the cycle.
Yes. The lymph coalesces into larger collecting ducts, And it finally empties into the subclavian veins near the chest, primarily via the thoracic duct, which is the largest lymphatic vessel, and the right lymphatic duct. This completes the fluid return cycle.
The pathology here, lymphedema, is a profound reminder of just how necessary this drainage system is.
Lymphedema is the chronic, localized swelling that's caused by compromised lymphatic drainage. It occurs when that delicate balance is shattered. The system just cannot remove the net filtered fluid and, critically, the high concentration of proteins that escape the blood.
And the proteins are the real problem.
They are. The protein-rich fluid accumulation leads to chronic inflammation, fibrosis, and hardening of the tissue. And secondary lymphedema, which is often seen after the surgical removal of lymph nodes during cancer treatment, really highlights the absolute vulnerability and necessity of this specific architectural network.
Okay, we have covered the smooth muscle regulator and the high and low pressure plumbing networks. Now, in part 7, we absolutely must address the specialized engine that drives this entire process, the cardiac muscle.
Cardiac muscle is a unique biological machine. It's essentially a hybrid that's optimized for relentless, rhythmic, sustained pumping. Structurally, it does resemble skeletal muscle. It's striated. It contains myofibrils organized into sarcomeres, complete with Z-discs, A-bands, and I-bands.
But the cellular architecture is totally adapted for continuous aerobic work.
Absolutely. Cardiac cells, or cardiomyocytes, are shorter, typically 100 to 120 micrometers long, and they are branched, forming this highly interconnected network. And if you look inside, you immediately notice their enormous energy requirements. They contain massive, numerous mitochondria, which reflects their almost exclusively aerobic metabolism.
So they need a constant supply of oxygen.
Constant. They cannot tolerate oxygen deprivation for more than a few minutes without sustaining significant, often irreversible damage.
The key structural difference that allows the heart to function as this unified, tireless pump is the intercalated disc. What are these complex structures doing simultaneously?
Intercalated discs are these complex, specialized junctional complexes, and they're found across the ends of adjacent cardiac cells where they abut. They are the single most ingenious piece of cardiac architecture because they achieve both mechanical force transmission and electrical coordination at the same time.
Okay, let's break down the mechanical linkage first. How do they prevent the cells from literally tearing themselves apart under that constant, intense pressure?
That mechanical anchoring is handled by two main components. First, you have fascia adherens. These are broad areas where the actin filaments of the terminal sarcomeres anchor directly to the plasma membrane. They are crucial for transmitting the contractile force from cell to cell along the longitudinal axis.
Okay, that's for end-to-end pulling.
Exactly. And second, you have desmosomes. These act like spot welds, anchoring the intermediate filaments and providing powerful mechanical resilience. They literally prevent the cells from separating during the massive systolic contraction force.
So mechanical integrity is maintained. Now what about the electrical syncytium, the rhythm of life itself?
That's the function of the gap junctions, which are strategically located in the lateral or side portions of the intercalated discs. They are electrical synapses. They allow rapid passage of ions and small signaling molecules directly between the cytoplasms of neighboring cells.
Just like in the single-unit smooth muscle.
Very similar concept. This instantaneous electrical coupling ensures that the depolarization signal spreads extremely quickly across the entire myocardium, allowing the heart to contract as one coordinated functional syncytium.
Okay, let's look at the contraction mechanism, specifically the calcium handling. The source material notes a difference in the internal structure, the arrangement of the T-tubules and the sarcoplasmic reticulum, the SR.
Right. In cardiac muscle, the invaginations of the cell membrane, the T-tubules, are actually larger than in skeletal muscle, but the arrangement with the internal calcium stores is different. Instead of the triads you see in skeletal muscle, which is one T-tubule flanked by two SR cisternate, cardiac muscle features dyads.
Dyads, so just one of each.
One T-tubule adjacent to only one SR cisternate. And this structural difference relates directly to the calcium mechanism, which relies on a dual source.
This is the famed calcium-induced calcium release, or CICR. Walk us through that cascade.
Okay, so CICR starts when the cardiac action potential travels down the T-tubules. This electrical signal opens voltage-gated Ci2+ channels on the T-tubule membrane, and this allows a small but critical amount of extracellular calcium to enter the cell cytoplasm. This initial calcium influx acts as the trigger.
So it's a trigger. What does that trigger calcium do?
The trigger calcium then binds to the ryanodine receptors on the membrane of the SR. This binding causes the massive, rapid release of internal calcium stores from the SR into the cytoplasm. This large increase in intracellular calcium concentration then binds to troponin C, initiating cross-bridge cycling and that powerful systolic contraction. It's a beautifully choreographed amplification system.
The actual duration of the cardiac contraction is also key to efficient pumping, and it's dictated by a very specialized action potential shape.
Yes, the cardiac action potential has a very long duration. We're talking hundreds of milliseconds. And this is due to a sustained plateau phase. This plateau is caused by the slow, sustained influx of calcium ions through those membrane channels we just mentioned.
So the calcium keeps coming in.
It does. And the sustained calcium presence ensures a prolonged contraction phase, which is physiologically absolutely essential.
Why is that prolonged contraction so essential, especially when you compare it to the really short, twitchy contractions of skeletal muscle?
The heart has to complete its full systolic ejection cycle, and the long refractory period caused by this plateau prevents the heart muscle from undergoing tetanus, which is a sustained, unfused contraction.
Which would be fatal.
Instantly. If the heart went into tetanus, it couldn't relax and fill with blood, leading to immediate failure. The prolonged action potential ensures that a proper contraction relaxation cycle is maintained for efficient pumping and filling.
The rhythm itself is intrinsic, generated by the pacemaker cells, but the autonomic system acts as the fine-tuner, regulating the rate and the force.
The autonomic nervous system provides the necessary modulation based on the body's demands. Sympathetic input, typically via noradrenaline acting on beta-adrenergic receptors, increases both the heart rate, which is chronotropy, and the force of contraction, inotropy.
How does it do that?
It achieves this by stimulating protein phosphorylation, which accelerates calcium release from the SR and speeds up the cross-bridge cycling. Basically, it makes everything happen faster and harder.
And the regulatory brake that slows things down when we rest?
That's the parasympathetic system, mediated by acetylcholine acting on muscarinic receptors. Its primary effect is to decrease the heart rate by acting on the pacemaker regions, the SA and AV nodes, slowing their depolarization rate and bringing the heart back to a more energy-efficient resting pace.
Finally, let's just revisit the vulnerability of this engine room. Because it is almost exclusively aerobic, its continuous workload makes it extremely susceptible to ischemia.
That is the tragedy of myocardial damage. Given the high metabolic demand, a lack of blood flow or ischemia rapidly leads to cell death. Cardiomyocytes are energy-demanding fragile cells.
For decades, we all believed that heart cells, once gone, were irreplaceable. The source material seems to update this understanding regarding regeneration.
It does. While the heart's regenerative capacity is extremely limited compared to, say, the skin or the liver, new evidence does suggest a slow, continuous turnover of cardiomyocytes. It's estimated at about 0.4% to 1% per year in healthy adults.
So it's not zero.
It's not zero. This low, continuous turnover suggests that the heart is not a completely static organ, and this is opening new avenues for research into therapeutic regeneration.
And the heart is always remodeling in response to stress, like chronic hypertension. This can be adaptive at first, but it often turns maladaptive.
That remodeling is critical to understanding chronic heart failure. In response to high blood pressure, the heart tries to adapt, often through hypertrophy. The individual cells enlarge, making the muscle wall thicker to generate more pressure.
Which works, for a while.
For a while. While it's initially compensatory, this physical and chemical remodeling can become maladaptive. It leads to altered cellular signaling, changes in the extracellular matrix, and eventually a less efficient, stiffened, or dilated ventricle, which results in chronic heart failure. Targeting these maladaptive remodeling pathways is a major focus in modern cardiac therapeutics.
This has been an incredibly detailed breakdown of the body's entire internal infrastructure. I mean, we started with the unique twisting architecture of that energy-efficient, smooth muscle cell, and we moved all the way through the high-pressure circuits, the critical exchange zone, the low-pressure return system, and finally, the relentless cardiac engine.
The key synthesis here, I think, really lies in the dual functionality of muscle. We saw the structural differences. The striated, fast-acting, troponin-regulated contraction of the cardiac muscle, which is necessary for powerful ejection. And you contrast that with the non-striated, calmodulin-regulated, energy-efficient latch state of smooth muscle, which is necessary for sustained arterial tone.
And we saw how these unique tissues translate into sophisticated macroscopic architecture. The elastic laminae in the aorta acting as a perfect hydraulic shock absorber to enable the Windkessel effect. The muscular arteries and arterioles precisely regulating resistance. And the specialized permeability of the 4 capillary types allowing tissues to filter or absorb or just protect themselves.
It is a system of constant unseen work. I mean, think of the seamless electrical synchronization achieved by the gap junctions in the heart, driving the flow, and then that subtle fluid balancing act dictated by starling forces in the capillaries, with the lymphatic system diligently mopping up the inevitable fluid overflow. This entire system operates through local, hormonal, and autonomic control without a single conscious command from you.
That brings us to our final provocative thought for you, the learner. We have described this perfect, tightly regulated machine. Now consider how subtle shifts in the control of that smooth muscle, the master regulator we started with, can manifest in these profound, debilitating disease states.
Right. Consider a disease like Raynaud's phenomenon. This isn't a structural disease, it's a functional one. A hyperreactive disorder of cutaneous vasoconstriction that's regulated entirely by the autonomic nervous system.
So the pipes are fine, but the control system is glitchy.
Exactly. The smooth muscle in the small arteries of the fingers and toes constricts abnormally and intensely, and it can temporarily shut down circulation entirely. It shows how the simple, functional misfiring of smooth muscle can override the system and cause painful local tissue ischemia.
Or you can contrast that with something like pulmonary hypertension. The pulmonary circuit, which is naturally a low-pressure system, is suddenly forced to operate at high systemic-like pressures. In response, the smooth muscle in the pulmonary arterial walls begins to thicken, to multiply through hyperplasia, and to remodel, fighting a resistance that just should not be there.
And this maladaptive remodeling leads directly to progressive right-sided heart failure. These conditions are profound exemplifications of the core truth we uncovered. The difference between seamless biological function and chronic destructive disease lies almost entirely in the disruption of the delicate cellular and tissue-level regulatory balance of the smooth muscle and cardiovascular structures.
The control is complex, highly effective, and deeply vulnerable to even the slightest maladaptation.
It is.
A stunningly engineered self-regulating machine, maintained down to the single-cell level. That was a deep dive into the inner architecture of circulation. Thank you for joining us.
Thank you. Keep learning.